I will share with you a quite difficult question, and we will try to answer. Are deep endometriosis and uterine adenomyosis two different phenotypes of the same disease? And I will share with you this paper published last month in Best Practice on Research Clinical Obstetric and Gynecology about the similarities and differences between endometriosis and adenomyosis. That is the program of my lecture. I will start with historical data, and it's a little bit back to the future. In fact, the first description of lesions named adenomyoma was provided by Carl von Rokitansky, and that was considered like a cystosarcoma. And only several years later, in 1925, 2 years before Sampson coined the term endometriosis, Frank created a name for mucosal invasion of the myometrium, calling it adenomyosis uteri. It was the first time that this term was used to explain the invasion of the mucosa in the myometrium. But really surprisingly, several years later, Sampson defined the cul-de-sac obliteration as extensive adhesions in the cul-de-sac, obliterating lower portion and uniting the cervix of the lower portion of the uterus to the rectum. And he called the term adenoma of the endometrial type. And it's only several years later that the term deep endometriosis was used. But finally, if you think at the literature, you see that von Rokitansky, Frankel, and Sampson already suggest the association between adenomyosis and endometriosis. Several years later, we published this paper, which has been cited very frequently, and for the first time, we call back the adenomyotic nodule of the rectovaginal septum, considering separate entities from peritoneal endometriosis and ovarian endometriosis. This term was used also when bladder endometriosis was considered. And we consider in the title that we should call this bladder endometriosis as bladder adenomyosis, and you can see clearly that the anterior wall of the uterus is also invaded by this tissue. And when we removed it, it has been removed by laparoscopy, we have this type of pathological finding, and that was published in 2000. In fact, if you show this slide to a pathologist, he will answer to you adenomyosis. That is exactly like adenomyosis of the uterus. That is for the story, historical data. What about the magnetic resonance imaging analysis? Coexistence of both disease. There was a nice cartoon published from the Johns Hopkins Hospital, when on this cartoon, clearly different type of endometriosis and uterine adenomyosis are on the same picture. And in fact, it's happened much more frequently than expected. And there are 3 papers by Charles Chaperon, our group, and Bazo. We clearly make the association between endometriosis and adenomyosis, considering that it's very frequent. In fact, in the description by all the lesions by Bazo, clearly in a part of the cartoon, the association between adenomyosis and endometriosis with attraction to the rectum or to the bladder were clearly defined. And remember that Bazo is not a gynecologist, he's a radiologist. And in the 2 papers following that by Charles Chaperon and by our group, we have analyzed the relationship between adenomyosis and endometriosis. Clearly, in our department, we have analyzed, retrospectively, the association between deep endometriosis with external uterocervical adenomyosis and junctional zone anomalies. This is a retrospective study. We select only the case with deep nodule more than 3 centimeters. It's a huge selection of the case. And in fact, the association between what we call external uterocervical adenomyosis, like in that case, exists in nearly 100% of cases, and the isolated association with junctional zone anomalies in nearly 30%. Thus, at that time, by this different publication, the association between deep endometriosis and adenomyosis could be advocated. If we agree with that concept, can we explain the mechanism? Are we able to identify common pathogenic features in both diseases? It's what we have done and published in the Journal of Clinical Medicine, identifying common pathogenic features in deep endometriotic nodule and uterine adenomyosis. And I will show you the result. In fact, we analyze the role of inflammatory cells and fibrosis in deep endometriosis and adenomyosis. And clearly, we select several cases. Thus, we took a biopsy from the endometrium from healthy women, utopic endometrium from women with deep endometriosis, from the nodule, what we call ectopic from deep endometriosis nodule, atopic endometrium from adenomyotic women, and the ectopic lesions from adenomyotic women, which mean a biopsy deep into the myometrium, revealing the endometrial glands. First analysis was the immunostaining by the CD41, which revealed the platelet activity, and obviously the platelet activity is reduced in ectopic in DEN and in ectopic in adenomyotic women when compared to the corresponding endometrium and corresponding also and when compared to the healthy endometrium. When we analyze the macrophage activity, there is a significant increase of the macrophage activity in ectopic tissue from DEN and ectopic tissue from the adenomyotic women. When we analyze the fibrosis, the fibrosis is obvious here on that picture that the fibrosis is significantly increased in ectopic endometrium as well in the deep endometriosis lesions as well in the adenomyotic lesions. And we go further to analyze the vascularization. And by analyzing the vascularization, we were unable to demonstrate any difference from the VEGF. But in fact, when we analyze, we see some difference in the macrovascular density and the, also in the maturity of the vessels. So that the macrophage activity was for us a concern that we would like to analyze deeper. And there was a study made by, in the lab of Professor Dolmans by Christina Stratopoulou. We evaluate the role of M2 macrophage on a culture promoting the endometrial cells invasion in uterine adenomyosis. In fact, if you can see here, the epithelial invasion capacity as well as the stromal invasion capacity. You see that when we put the epithelial cells in culture, in the presence of M2 macrophage, there was an increase of the invasion capacity in the adenomyotic women, more significant than in the women from the control group. That was the same result for the stromal capacity. It means that the M2 macrophage, the macrophage, activated macrophage, play a role in the invasion process. And it will be published very soon. It's accepted in the press. Of course, we have to analyze the role of oxidative stress. We have forget for a long time the role of iron, which produce oxidative stress, and on the other side, the erythrocyte can stimulate the macrophage, activate the macrophage, which are for the peritoneal cavity responsible for the cleaning of the peritoneal cavity. But when they are activated or overwhelmed, they play a role in the oxidative stress and in the invasion of the tissue. This cartoon will be also published very soon, and it explains the role of the macrophage. They are able to phagocyte, of course, heme, and to be the cargo of the hemosiderin, but don't forget that they are also responsible to initiate the oxidative stress by the significant increase of the reactive oxygen species. And when these macrophages are activated, they are able to promote the invasion, the adhesions, the angiogenesis, and the activation of the inflammation. Just imagine that in the future, we may influence the macrophage activity by depleting the macrophage activity. We have maybe an option to influence the development of the disease. And we are not alone to have this idea. A lot of people have this idea, and I would like to go with you with this excellent paper and review published by the team of Christian Becker in a human reproduction very, very soon. And you see that he focused on the small extracellular vesicles, which are the cargo for the microRNA. What happens? In fact, the red blood cells, all the cells are able, and the endometrial cells too, are able to release the small extravesicular vesicle, which contain the microRNA. But they are able also to activate the macrophage, which afterwards are able to release themselves several extracellular vesicles with the microRNA, influencing the apposition, the transformation, and the angiogenesis to finally provoke the endometriotic lesions. Apoptose, autophagy, and progesterone resistance also implicated in both disease. As said by Marie-Madeleine, in progesterone resistance has been proved in adenomyosis, and Hugh Taylor has proved in endometriosis. Autophagy and apoptose in both diseases should be evaluated. And finally, the experimental model. There are three papers which has been extensively discussed with Philippe Connings. There's the thesis of Olivier Donnez and the baboon model. In fact, when we have a transplant in the baboon, endometrium, junctional zone, and myometrium, we think to reproduce the deep endometriotic lesions. But in fact, we have reproduced deep adenomyosis, uterine adenomyosis, like it's proved. In the next picture here, you see very well the big nodule here with some invasion of the glands through the bowel wall. So that, in fact, some experimental model reproduce very well the adenomyosis, which is quite similar to deep endometriosis. But it's just at that time for the baboon model and hypothesis. So that, no. In fact, we have to come back to several ideas. This paper was published by the lab, and of course, in case of uterine adenomyosis, a majority of the authors are in favor of what we call TIAR, the endometrial invasion responsible for adenomyosis. Or the second hypothesis is the presence of Mullerian remnant into the myometrium. But from the recent publication of the lab, we have to pay more attention to the stem cells, which are present in the endometrium and in the junctional zone, and these stem cells present into the myometrium could be responsible of the adenomyosis process and be responsible by retrograde menstruation of the presence of stem cells able to create endometriosis, so that finally the same origin, the stem cells, could be at the same origin of the two phenotypes. So that there are differences and similarities, and I had two slides about the treatment, management, medical management. And in fact, when you analyze the possibility of medical management for adenomyosis and endometriosis, they are the same. We have to target estrogens, target inflammation, NF-kappa B and cytokine, ROS, apoptose and autophagy, MT regulators, and targeting angiogenesis. But all these pathways are under clinical experimental evaluation, and in fact, there are so many pathways explaining uterine adenomyosis and deep endometriosis that it remains a long way to find a solution. Finally, are there two different phenotypes of the same disease? I guess yes, but further evidence is needed, and maybe the artificial intelligence will give you the answer very, very soon. Thank you for your attention.